6A provides a framework for specifying drug substance and drug product properties. Particle size is a critical parameter within this guideline, particularly for solid oral dosage forms, because it can significantly affect the drug's performance and quality. Here's why particle size is so important in ICH Guideline Q6A. Dissolution rate. The dissolution rate of a drug is influenced by its particle size. This is particularly important for poorly soluble drugs, where the dissolution rate is the rate-limiting step for absorption. Bioavailability. Particle size can directly impact the bioavailability of a drug. Enhanced dissolution can lead to improved bioavailability, meaning more of the drug is available in the bloodstream to exert its therapeutic effect. Dosage uniformity. Consistent particle size distribution ensures uniformity in the content of the active pharmaceutical ingredient in each dosage unit, for example, a tablet or a capsule. This uniformity is crucial for ensuring that patients receive the correct dose. Manufacturing process. Particle size affects various manufacturing processes such as mixing, granulation, and compression. Consistent particle size can lead to more efficient and reproducible manufacturing processes, reducing variability and improving product quality. Stability. Particle size can influence the stability of the drug product. Smaller particles may be more prone to chemical degradation due to their larger surface area, which can be addressed by optimizing particle size during formulation. Formulation and process robustness. Understanding and controlling particle size is essential for the robustness of the formulation and manufacturing process. It helps in developing scalable processes and maintaining product quality throughout the product's life cycle. And finally, regulatory compliance. Compliance with ICH Q6A guidelines regarding particle size ensures that the drug product meets international quality standards. This compliance is crucial for regulatory approval and market access in different regions. Let's move on now to an overview of the role of zeta potential in pharmaceuticals. Zeta potential is defined as the electrokinetic potential in colloidal dispersions. It is a measure of the electrical charge on the surface of particles and is crucial for predicting the stability of pharmaceutical suspensions. Generally speaking, high zeta potential, whether positive or negative, indicates good stability, as it means the particles will repel each other and remain dispersed rather than aggregating. Factors that affect zeta potential include the pH and ionic strength of the medium. Zeta potential is also crucial in the realm of drug delivery. Nanoparticles with specific zeta potential values can be used for targeted drug delivery. This helps in reducing side effects and improving the efficacy of the drug by ensuring it reaches the intended site of action. A familiar example is lipid nanoparticle-based mRNA vaccines, because the uptake of the particles by the cells depends on the surface charge of both the particles and the cell walls. Let's look at how the guidelines address zeta potential. While zeta potential may not have its own specific guideline, it is a critical parameter across various ICH guidelines, impacting drug stability, quality control, and regulatory submissions. Let's start with ICH Q6A, which covers specifications for new drug substances and products, particularly chemical substances. Zeta potential plays a crucial role here, as it impacts particle size distribution and the surface charge, both of which are critical for ensuring drug stability. Next, we have ICH Q8R2, which focuses on pharmaceutical development. In the context of Q8R2, zeta potential helps in understanding and controlling the stability and performance of nanoscale drug delivery systems. Moving on to ICH Q9, which deals with quality risk management. Zeta potential is integral to risk management as it affects the stability and efficacy of drug products, making it a key factor in risk assessment and control. Finally, ICH Q10. This outlines a model for an effective pharmaceutical quality system. Consistent measurements of zeta potential are essential for quality control, ensuring that the product remains stable and performs as expected. Let's move on now to a brief description of the fundamentals of light scattering as it relates to measuring particle size and zeta potential using instruments such as the better size BeNano series. To understand light scattering, we first need to grasp the nature of light and how it interacts with matter. We'll use the wave theory of light, which states that light consists of two oscillating fields, an electric field shown in red and a magnetic field shown in blue. These fields fluctuate like ripples on the surface of water, and the wave propagates in a specific direction, such as from a laser source. Consider an electron positioned within this fluctuating electric field. The electron, which carries a negative charge, moves in response to these fluctuations. This movement or acceleration of the electron generates its own electric field. And according to physics, an oscillating electric charge creates both an electric and a magnetic field, thus producing another electromagnetic wave. This new wave propagates in all directions. In a light scattering experiment, a photodetector is placed to capture a narrow portion of the light scattered by electrons, such as those in nanoparticles. When you have multiple nanoparticles, each at different locations, the photodetector detects light from all these particles. The key to understanding the scattered light is the phase relationship between the waves emitted by each electron. By analyzing these relationships, we can extract information about the motion of the particles. This motion could be random due to diffusion, which helps us estimate the apparent size of the particles using dynamic light scattering. Alternatively, if the particles move steadily due to an electric field, we can estimate their charge or zeta potential using electrophoretic light scattering. The better size BeNano offers both dynamic light scattering and electrophoretic light scattering options. Let's start with an overview of dynamic light scattering, or DLS. Dynamic light scattering, also known as photon correlation spectroscopy or quasi-elastic light scattering, measures fluctuations in the intensity of scattered light due to Brownian motion of particles. In the animation, you can see three particles moving randomly. The electrons in each particle emit light in all directions, with a phase relative to the incident light based on the particle's position in space. Consequently, the detected composite signal fluctuates randomly because the relative positions of the particles are continuously changing randomly. The overall intensity of the light sensed by the photodetector changes in a random way, with the characteristic time frame of this randomness matching the characteristic time frame of the particle's Brownian motion. In a DLS experiment, a laser beam is directed through the sample, and the scattered light is detected at a fixed angle, often 90 degrees. Some instruments offer other angles depending on the model. A correlator performs a function called autocorrelation, which enables estimation of the diffusion coefficient. And from the diffusion coefficient, the hydrodynamic diameter and size distribution of the particles in the sample can be determined. Autocorrelation is a technique used to characterize a random signal and estimate its characteristic time frame. Let's examine this graph, which displays two examples of randomly fluctuating signals. The blue line represents slow diffusion, typical for large particles, while the brown line represents fast diffusion, expected from small particles. Visually, you can see a clear difference in the time frame over which the randomness changes. We can exploit this difference using correlation. Now, the concept is straightforward. If you take a signal and then look at it a certain amount of time later, the new value of the signal will depend somewhat on the original value. This dependency is called correlation, and since the signal is compared with itself, it's called autocorrelation. For a random signal, there are two extreme limits in correlation. First, when the signal is compared to itself at the same time, there's a 100% correlation because the signal is unchanged, so the correlation function starts at 1. Second, if the signal is compared to itself after a long time, there's no correlation, so the value is 0. Typically, a gradual decay occurs between these extremes. The upper trace on the graph shows how the signal is sampled over specific time intervals, illustrating the correlation between adjacent points. Repeating this process over many parts of the time signal creates a decaying curve. For faster diffusing particles, this decay happens more quickly. The decay is approximately defined by an exponential function, e to the minus dq squared t, where d is the diffusion coefficient and q is the scattering vector. The instrument measures this function through light scattering to estimate the diffusion coefficient, which in turn requires knowing the scattering vector. The diffusion coefficient relates the particle's physical properties, and the scattering vector accounts for the instrument's optical geometry. The Stokes-Einstein equation relates the diffusion coefficient to the hydrodynamic diameter, d sub h, through the Boltzmann constant, the sample temperature T, controlled by the instrument, and the viscosity of the medium, eta. The scattering vector depends on the medium's refractive index, n, not to be confused with the refractive index of the particles, the scattering angle in air theta, and the laser wavelength in air lambda sub 0. When the correlation function is plotted on a geometrically spaced time axis, the exponential decay appears S-shaped, as seen with instruments like the BeNano. Electrophoresis refers to the movement of charged particles in response to an electric field. Traditional electrophoresis uses a constant direct current, or DC, electric field, whereas laser Doppler electrophoresis applies a slowly reversing square wave electric field. Despite the square wave nature, this can essentially be considered a DC electric field. In the BeNano series of instruments, this mode is known as the slow field reversal mode. The detected signal depends on particle movement, combining both diffusive and linear motions due to the applied electric field. As particles move linearly, the phase difference between incident and scattered light changes linearly, producing a Doppler shift frequency. The overall signal is a sum of these Doppler shifts from all particles. To analyze these signals, we use a Fourier transform to obtain a distribution of frequencies. Using the scattering vector, we can determine the velocity distribution of the particles. Knowing the electric field, we can calculate the electrophoretic mobility, which measures the particle's response to the electric field. The zeta potential, an indicator of particle surface charge, can be estimated using theoretical models. Hence, electrophoretic light scattering provides us with particle surface charge information. As with dynamic light scattering, a laser beam is passed through the sample. However, photodetectors can't capture absolute motion, only relative motion of particles. For electrophoresis measurements, a reference is needed, which can be achieved with a second laser beam acting as a stationary particle reference. However, a stationary particle reference can't distinguish particle direction, only providing us with the charge magnitude. A moving reference, achieved by reflecting the reference beam off a moving mirror, addresses this by shifting the reference beam frequency, allowing for the determination of electrophoretic mobilities or zeta potentials via the Fourier transform. And when diffusion dominates, resolution of the electrophoretic motion becomes more difficult. This can be the case with weakly charged particles, high salt concentration media, or nonpolar media. I developed phase analysis light scattering, PALS, to overcome the limitations of the laser Doppler method. It uses the same hardware, but applies a faster alternating electric field, the fast field reversal mode. PALS measures periodic changes in the optical phase corresponding to particle motion in an alternating electric field. And by collecting and averaging this phase information synchronously over many cycles of the applied electric field, any noise in the signal, such as due to diffusion, averages to 0. This is the reason why PALS is more sensitive and more precise than LDE. The average electrophoretic mobility is readily calculated from the averaged phase function. However, it doesn't provide a distribution of values. The BeNano instruments combine slow and fast field reversal modes to provide both a distribution and an accurate estimate of electrophoretic mobility and zeta potential, enhancing measurement accuracy and applicability. Let me wrap up with the key takeaways. Particle size is crucial in pharmaceuticals. Smaller particles improve dissolution rates and bioavailability, ensuring that more of the drug reaches the bloodstream effectively. Consistent particle size also guarantees dosage uniformity, which is vital for patient safety and efficacy. Additionally, optimizing particle size enhances manufacturing efficiency and stability, leading to higher quality products. Zeta potential is key to predicting stability in colloidal suspensions. It's also vital in drug delivery, helping create targeted systems that reduce side effects and improve efficacy. And the BeNano series is the latest generation of nanoparticle size and zeta potential analyzers developed by BetterSize to help you develop innovative pharmaceutical products that follow ICH guidelines, ensuring that pharmaceutical products meet international quality standards. This compliance is critical for regulatory approval and market access, supporting the delivery of safe and effective medicines. With that, I'd like to thank you for your attention and now hand it over to Jubin, who will demonstrate how the BetterSize BeNano is an essential tool for analyzing the particle size and zeta potential of many of today's complex nanoparticle-based pharmaceutical formulations.
Thank you, John. I'm excited to talk about the innovative features of our BeNano series, focusing on its applications in size and zeta potential analysis within the pharmaceutical industry. Over the next 20 minutes, we will explore how the BeNano can significantly enhance the work in drug formulation, stability studies, and more. Let's begin with an overview of the BeNano system. The BeNano is equipped with 3 key technologies that provide a wide range of measurement parameters. Dynamic light scattering (DLS) offers measurements like the average size, polydispersity index (PDI), and size distributions by intensity, number, and volume, as well as rheological properties essential for nanoparticle characterization. Electrophoretic light scattering (ELS) using techniques such as phase analysis light scattering and laser Doppler electrophoresis measures average zeta potential, pH change, and zeta potential distributions, providing insights into colloidosis stability. Static light scattering (SLS) determines molecular weight and the second virial coefficient, which are crucial for understanding drug molecular interactions and stability. In the pharmaceutical industry, the BeNano is instrumental in enhancing users' understanding of materials and addressing critical challenges. In this talk, I will highlight 3 key benefits of the BeNano. First, its ability to detect even small aggregates, which is essential for ensuring drug safety and efficacy. Second, its capability to monitor size changes over time and under varying temperatures, aiding in the evaluation of formulation stability. And third, its zeta potential measurements, which provides insights into drug interactions with carriers, a critical factor in optimizing drug delivery systems. Let's dive into the determination of aggregates. Aggregates detection is not only a quality control measure, but also a regulatory requirement due to its critical impact on drug safety, efficacy, stability, and compliance. Aggregates can trigger immune responses, leading to immunogenicity and adverse reactions, potentially compromising patient safety. They can also reduce bioavailability and alter pharmacokinetics, diminishing the drug's therapeutic effect. Furthermore, aggregates signal physical instability and chemical degradation in formulations, posing long-term stability risks. To meet regulatory standards, pharmaceutical products must limit aggregate levels, requiring regular monitoring to ensure safety, efficacy, and compliance through the product's life cycle. So detection and quantification of aggregates are challenging due to their size variability and low concentrations. The BeNano enhances the conventional 90-degree detection method in DLS analyzers by employing a sophisticated 173-degree backscattering system. This advanced design allows for intelligent adjustment of the detection position through the lens movement, optimizing performance based on different sample types and concentrations. This technology offers higher detection sensitivity, enabling particle detection in samples with concentrations as low as 0.1 ppm, and supports a wider concentration range, handling up to 40% weight per volume by reducing multiple scattering effects. It also minimizes the dust interference, ensuring accurate and reliable measurements even in challenging environments. BeNano's backscattering technology provides a versatile, robust solution for particle size analysis across diverse sample concentrations, making it highly valuable for pharmaceutical applications. Here's an application of BeNano in a study involving DNA, demonstrating how gold nanoparticles can be utilized in pharmaceutical applications, particularly for detecting the Omicron variant DNA sequence. The process shows that when the DNA sequence of the Omicron variant binds to the gold nanoparticles detection probes, it leads to the aggregation of gold nanoparticles. This aggregation is critical as it produces a strong photoacoustic signal on a biosensor, which enables the rapid detection of DNA. This strong PA signal is crucial for detecting the presence of the target DNA sequence quickly and accurately. To explore the aggregation behavior of gold nanoparticles, researchers in this study incrementally added different concentrations of magnesium sulfate to a 0.1 nanomolar gold nanoparticle solution. This addition triggered the aggregation of gold nanoparticles, which was then monitored through the changes in the UV visible absorption spectrum. The results showed a decrease in absorption at 521 nanometers and a shift towards the higher absorption at longer wavelength as the magnesium sulfate concentration increased from 0.1 millimolar to 0.7 millimolar. Additionally, the hydrodynamic size of the gold nanoparticles was measured using DLS. The data indicated that the average hydrodynamic diameter of the gold nanoparticles grew from 37 nanometers to 136 nanometers as the aggregation progressed. The hydrodynamic size distribution graph reflects that the size increased with the distinct pins representing the different concentrations of the magnesium sulfate. Overall, this example demonstrates the effectiveness of using gold nanoparticles in conjunction with the BeNano to monitor a nanoparticle aggregation, a key process in the development of diagnostic tools for detecting viral DNA sequences. In the third part, we will explore the critical reasons for monitoring size changes over time and under varying temperature conditions. Researchers monitor particle size for several crucial reasons. First, it's essential for stability assessment, as changes in particle size over time can affect a product's shelf life and performance. Monitoring helps detect deviations early, ensuring quality control. And second, particle size is sensitive to temperature fluctuations, and tracking these changes during storage and transport ensures the material stability until it reaches its final destination. Third, observing particle size variations helps identify degradation mechanisms enabling improvements in formulations and extending product lifespan. Lastly, regulatory compliance mandates documentation for particle size changes, ensuring adherence to industry standards and maintaining both legal compliance and customer trust. The BeNano software offers the flexibility to set a time delay, which allows users to specify the interval between each instrument. This functionality enables the detailed observation of trends in both particle size and zeta potential over time, providing insights into the dynamic behavior of the sample. For instance, users can configure the software to take a measurement every 3 minutes, resulting in a total 10 measurements over 30 minutes. This setup allows for the continuous monitoring of size changes, which is crucial for understanding how biomolecules behave over extended periods. In this slide, we will explore an example of a target drug delivery system utilizing biometal organic frameworks, abbreviated as BioMOFs. This framework serves as a foundation for developing highly stable and effective therapeutic nanodelivery systems. The process begins by coating BioMOFs nanoparticles with polydopamine at a pH of 8.5, resulting in MP nanoparticles. These MP nanoparticles are then loaded with the chemotherapy drug doxorubicin, a widely used cancer treatment, to enhance the biocompatibility and targeting capabilities. The surface of these MP nanoparticles is modified with hyaluronic acid, creating MPH nanoparticles. This modification is crucial as it allows for the drug delivery system to target specific cells more effectively, potentially reducing side effects and increasing treatment efficacy. To evaluate the stability of both MP and MPH nanoparticles under conditions that mimic the human body, researchers incubated them into different environments, PBS and FBS. PBS simulates the physiological conditions, while FBS represents the plasma environment found in the bloodstream. These conditions were maintained over a 72-hour period, during which the particle size of the nanoparticles was carefully monitored. The results are compelling. Through the 72-hour incubation period, both MP and MPH nanoparticles demonstrate the remarkable stability. Their sizes remained consistent, showing no significant changes, which is a strong indicator of their resistance to the destabilizing effects of salt ions and proteins commonly found in the body. This stability is critical to ensuring that the drug delivery system functions as intended when administered to patients. A stable particle size means that the nanoparticles are less likely to aggregate or degrade, which could otherwise compromise the effectiveness of the treatment. In addition to time trend measurement, the BeNano offers temperature trend measurement across a range from -15 to 110 degrees Celsius. A key benefit of this function is its ability to assess protein stability. By gradually altering the temperature, users can identify the denaturation point of proteins, a crucial indicator of formulation stability. A higher denaturation temperature generally indicates a more stable protein formulation, making this feature essential for optimizing and validating biopharmaceutical products. Moreover, the program temperature change function is highly effective for simulating accelerated aging processes. By applying elevated temperatures, users can speed up the aging of samples, reducing the time needed to observe long-term stability effects. This capability is useful for industries that need to understand the lifespan and durability of materials or formulations under stress conditions. In this slide, we will be discussing an example of lysozyme. Lysozyme was measured at a concentration of 30 milligrams per ml, with the temperature gradually increased to assess its impact on particle size. As you can see from the graph on the left, the Z-average size and scattering intensity of lysozyme remained stable up to 50 degrees Celsius. This stability indicates that lysozyme maintains its structural integrity at these temperatures. However, when the temperature rose above 55 degrees, a significant change was observed. The Z-average size increased notably, suggesting that the lysozyme molecules began to aggregate. This is further supported by the size distribution curves on the right. As the temperatures above 55 degrees, the distribution shifts dramatically, indicating the formulation of larger aggregates. These changes can be attributed to the denaturation of lysozyme at higher temperatures. Denaturation is a process where the protein loses its native structure, leading to aggregation as the molecules stick together. This is a common issue with proteins and underscores the importance of studying their temperature sensitivity. In the final part of this talk, let's shift our focus to zeta potential and explore how it can serve as an indicator of drug interactions. Zeta potential is commonly used to study the isoelectric point, IEP, and surface absorption of particles in liquid media, serving as an indicator for comparing the electrostatic dispersion stability of different samples. It's not an inherent property of suspended particles. It depends on the properties of the particles and the medium, as well as their interactions at the interface. For many biopharmaceutical examples, a notable change in the zeta potential of nanoparticles upon drug loading often suggests an interaction between the drug and the carrier material. This interaction may result from ionic interactions or the formation of a complex between the drug and the carrier. Understanding these changes is essential for optimizing drug formulations and ensuring that the delivery system functions as it intended. This example shows an application of calcium silicate nanocomposite, abbreviated as MCN, as a drug delivery system, and specifically after being loaded with indocyanine, ICG. MCNs are advanced materials that combine calcium silicate with nanoscale reinforcements, enhancing their properties and performance. The particle size and zeta potential of MCNs were measured before and after loading with ICG. The result shows that the particle size increased from 180 nanometers to 143 nanometers with a narrow distribution range, indicating a uniform size after drug loading. Additionally, the zeta potential shifted from minus 30 millivolts to minus 8 millivolts, signifying a change in surface charge. These changes in both size and zeta potential suggest a significant interaction between the loaded drug ICG and MCNs. This interaction is primarily due to hydrogen bonding between the ICG molecules and these silanoid groups on the surface of the mesoporous walls of the MCNs. The sulfonate groups in ICGs with their oxygen atoms serves as strong hydrogen bond acceptors, allowing each ICG molecule potentially neutralize multiple silanoid groups on the MCNs. This extensive hydrogen bonding leads to the observed reduction in zeta potential after ICG loading. Overall, this example demonstrates how the loading of a drug onto a nanocomposite carrier can be monitored and analyzed through the changes in size and zeta potential using the BeNano, providing the insights into the nature of the interaction between the drug and its carrier. These are the key takeaways from this presentation. The BeNano system excels in nanoparticle characterization, particularly for drug formulation, stability analysis, and aggregates detection through advanced backscattering technology. Additionally, its ability to monitor size changes under varying conditions and measure zeta potential provides crucial insights into biomolecule stability and drug carrier interactions. Thank you for your attention throughout this talk. We hope these insights into the BeNano system's capabilities have been valuable. Should you have any further questions or need additional information, please feel free to reach out.
Thank you both for providing your insights today. That was great. And thank you to our attendees too, who've been giving some very positive feedback throughout the presentation. It's great to see. I also want to remind all attendees that there's the handout section, which you can download various handouts, one of which is sign up for Better Sizes next webinar, which takes place on the 13th of November. So without further ado, we're going to move on to the Q&A section. Thank you for posting your questions. John, if I start with you first, you explained that dynamic light scattering measures the Brownian motion of particles. Could you describe some of the challenges or limitations of DLS when dealing with highly polydispersed samples or complex formulations?
Yes, thank you for the question. So one of the main challenges with DLS when working with either highly polydispersed or complex formulations is that the technique can struggle to accurately distinguish between particles of very different sizes. There are two main factors that lead to this. The first is that the technique is inherently sensitive to the intensity of the light scattered by the particles, and that varies according to the size by about a power of 4. So very large particles will dominate the signal compared to those from small particles. And also the selection of the angle that you're making the measurement at impacts the scattering that it's able to access. So you have to be, with an instrument like the BeNano, you can select different angles, and that helps you see if there is a contribution from very large particles when you're looking also for very small particles. The other challenge is, when I showed the example of an autocorrelation function, it's just a single curve. And somehow the instrument has to extract from that a distribution of curves. There's actually, every particle contributes its own decay. And what you're seeing is a composite of all of them. And mathematically, that's a very difficult thing to do. And it requires, even for the simplest models, it requires knowledge of the optical properties of your particles. So that's why the instrument will ask you to enter information such as the refractive index of the particles and their absorbance. And very often you don't really know that. And the instrument also assumes that those particles kind of have a uniform refractive index. But if you have some with complex nanoparticles, where you either perhaps you have a mixture of materials dispersed within the particle, or you have a layer of one material on another, then the assumptions that the model has to estimate a size distribution aren't met. So you have to just be cautious about how you interpret those results. However, having said that, it really does depend on what you're trying to do. Do you need to know the particle size, or are you trying to control a process? So if you've developed a formulation and you have manufactured a number of batches and you've collected data that show whether a particular batch meets stability requirements or not, then if you apply the ICH guidelines and you use quality by design methods, you can identify if particle size as measured with a particular instrument is a critical quality attribute. And once you've done that, you don't really need to know what the actual particle size is. What you're looking for is the range over which a particular particle size distribution that you get from your formulation corresponds to something that you know is likely to be stable or unstable. And in fact, so in principle, you can actually just use the correlation function for that too. What's important there is that you would develop a method and you have to very precisely define the instrument that you're using, the sample preparation details, the instrument's method that you followed, all the parameters that you entered into the instrument, and so on. And if you specify those things, you can still very usefully use dynamic light scattering, even for complex and highly polydispersed samples.
Fabulous, thank you. And John, just staying with you for one more question. Mm-hmm. You highlighted the importance of zeta potential in predicting the stability of pharmaceutical suspensions. Could you discuss how variations in pH and ionic strength might affect zeta potential and consequently the stability of a drug formulation?
Yes, so zeta potential can be highly sensitive to both the pH and the ionic strength. Not always. It does depend very much on the nature of the particles and the medium that they're in. And both of those will influence the surface charge of the particles. So Jabin mentioned about measuring the isoelectric point, for example. He covered that very well about the importance of determining the isoelectric point. And so one use of the instrument might be, and going back to what I'm saying about critical quality attributes and using the instruments, even if you're not necessarily after a specific number for the zeta potential, is you can say, okay, we know that the isoelectric point has to lie between a certain range of pH, and you can use your instrument to then test a particular batch of product and determine that. Now, I do want to caution, though, that it's a very common belief that a high zeta potential always means stable and a low zeta potential always means unstable. And that's not true. Okay, it does depend very much on the nature of the particles. There are systems where even when the zeta potential is pretty much non-existent, the particles are very stable. Proteins are an example of that. Polymeric systems, and sometimes the zeta. And sorry, let me just back up. And the zeta potential assumes, or the calculations that the instrument does to estimate the zeta potential, makes assumptions about the nature of the particles. But you can find those assumptions in any good textbook on colloid science. And most complex nanoparticles actually don't meet those assumptions of the models. So the zeta potential that the instrument presents to you is useful, but it isn't necessarily correct because you're not using the right model, but it's still valuable. And in the case of isoelectric point, 0 millivolts for zeta potential corresponds to a 0 electrophoretic mobility. So in fact, you don't need to know the zeta. It doesn't matter if you're not using the right models to do the calculation, because you're looking for 0. And that will be true from the mobility as well, which is the fundamental measurement.
Fabulous, thank you. And now moving to Jabin. I've got a question here from someone who works with lipid nanoparticles. Could you please clarify if your device is capable of characterizing lipid nanoparticles and what specific information it provides during the analysis?
LNP is one of the typical applications of the BeNano. It allows for rapid analysis of different nanoparticles to optimize formulations, such as PLGA nanoparticles, liposomes, and ionizable LNPs. So the size and polydispersity of LNP are critical quality attribute, and they are highly dependent on the preparation processes, requiring precise control and customization. And on the other hand, stability can be evaluated by monitoring the changes in particle size and polydispersity, as I mentioned in the presentation. BeNano can provide information on size, distribution, and zeta potential, helping to evaluate the stability, surface charge, and thermosensitivity. It also enables the LNP researchers to streamline both R&D and QC processes, and increasing sample throughput. Through DLS and ELS, BeNano can detect aggregation and monitor particle behavior, making it valuable for drug delivery, gene therapy, and vaccine development.
That's fantastic, thank you. And staying with yourself, we've got someone else whose sample is dispersed in a high ionic strength medium. Would it be necessary to dilute the sample before measuring its zeta potential?
I get it. That's a good question. So, as we know, the high ionic strength can cause compression of the electrical double layer around the particles, which may reduce the zeta potential. Dilution with a lower ionic strength can help restore a better defined double layer, improving the reliability of the zeta potential measurement. However, it is essential to ensure the dilution does not alter the particle interactions or change overall system behavior. And to be specific, in a high ionic strength medium, there's another thing to keep in mind. The added salts increase the ionic strength, which shields the particle surface potential, decreasing the zeta potential. And in extreme cases, the zeta potential can approach zero. And BeNano has a technique called phase analysis light scattering. I believe John has introduced very well. It is effective at measuring zeta potential, even for samples with very low electrophoretic mobility in high ionic strength conditions. While dilution can, to some extent, help reduce ionic strength, BeNano's pulse techniques allows for accurate measurements without extensive sample dilution.
Fantastic, thank you. Now we've got some questions that I'm going to open up to both of you. The first one is it possible to determine the concentration of nanoparticles using the instrument? And what are important factors in that?
In principle, it is. I'm not familiar enough with the instrument to know whether that is implemented or not.
That's no problem. We can move on to another question. Is how are samples prepared for analysis?
Huh. That's quite a broad question. So let's say in either case, typically, a lot of pharmaceutical formulations will be quite opaque. So if they've got high solids concentrations, for example. So the challenge there is the dilution. And it's very important that you dilute the particles into a medium which is either identical to the one they're already in, or as closely matches it as possible. Because sometimes it's not available, because often you won't make sort of the equivalent of a placebo version. And the medium might have various surfactants and electrolytes and other materials added, and they all will contribute to the interfacial properties of the particles in that medium. So you also have to appreciate when the particles are in a concentrated system, if the particle interactions are actually different than they are when they're dilute. So that's where I would recommend making measurements as a function of dilution and see if you see any trends. And this applies both to zeta potential and to particle size. And the other aspect, another aspect related to that is the presence of typically dust and other large scattering materials. And that's very well-known good practice for dynamic light scattering. So it's with pharmaceutical systems, typically they're going to be fairly dust-free anyway.
Great, thank you.
I can have some adds on. So sample preparation for DLS and zeta potential analysis in the BeNano involves diluting the sample to a suitable concentration, as John explained, to prevent multiple scattering, ensuring the medium's refractive index is known, and removing the aggregates through filtration or centrifugation. Limitations include the need for proper dilution to avoid multiple scattering, which can distort results, and ensuring that the sample remains stable after preparation, which is very important. So to wrap up, missteps in preparation for either VLS or ELS will affect the accuracy and repeatability.
That's great, thank you. And is it possible to take the size and zeta together in the BeNano?
Okay, I can answer that question. The answer is yes. The BeNano allows for measurements of both size and zeta potential. By integrating the DLS for size measurement and ELS for zeta potential analysis, the BeNano can provide a comprehensive characterization of nanoparticles in a single instrument. And this combined approach helps in understanding both the physical dimensions and the surface charge properties, which are crucial for many users for predicting the stability and particle behaviors in different formulations.
So I just want to clarify, because it wasn't clear in the question, if the question was asking if you can do that at the same time, or if it was just asking generally, can the instruments do both? So it can't make the measurements simultaneously, but you can run one after the other, and you can do the sizing in the presence of the electrodes that would be in the cuvette, or if you're using the folded capillary. So yes, it can be done on the same instrument. Also, if you have a model that offers both, because I believe there are variations of the BeNano instrument, some of which only offer the zeta potential, some of which only offer the size. So it does depend on which instrument you've purchased.
Fabulous, thank you. I think that comes to the end of our questions today. Do either of you have any final thoughts or insights you would like to share?
I'd just like to thank you for the opportunity to present, and thank you to everyone who's listened.
Yeah, thank you for everyone attending this webinar. Please feel free to contact us after this presentation.
Yeah, and I'll follow on by also thanking everyone who's attended today and thanking John and Shuabin for their amazing presentation. I hope you've gained lots of insights from it. Like I've said, handouts are available. And yeah, have a great rest of your day, and hopefully see you in the next webinar.
Thank you.